Okay, so I think we can just maybe 1 more minute and then we start. There are about 70 people already. Hi, Anne.
Hi, everyone.
Hi.
Justin.
Hi. I thank God. How are you?
Good, good. Yourself?
Right. Same. I'm good. Thanks.
Okay. We decided to give people 3 minutes from the start time. So I think 3 past the hour, then we just start.
Good afternoon, everyone.
Hi, Shiresh. Okay, I think we can start basically. So my name is Thank God Ebenezer, and I'm a bioinformatician at the European Bioinformatics Institute. So basically, one of the things I do is mainly to write computer programs to analyze proteins that comes through the Universal Protein Resource database. So today, we're going to be talking to Ross about the African BioGenome Project. And on the agenda, I'm just going to post the agenda on the chat box so people can see this as well. So the first item on the agenda mainly is to introduce members of the AfricaBP committee. So maybe I'll just hand this over to Anne to chair the session, who is the chair for the African BioGenome Project, and then we can commence. Over to you, Anne, please.
Thank you very much. Thank God. So the African BioGenome Project will be taking you through it so that you can be able to know exactly what is envisioned and what is the plan for this. My name is Anne Wigai. I'm a Kenyan professor of genetics, working in Jomo Kenyatta University of Agriculture and Technology, which is based in Kenya. I am the current chair of the African BioGenome Project. And we have a number of co-chairs, and one of them is Thank God. You can wave.
Hello.
Thank God is based in the European Bioinformatics Institute. He's in the UK. And then we have Catherine Zimoyo, and she is based in Biosciences East and Central Africa, Beka Iri, Nairobi. And we have Josiah Mutuku, who's a co-chair also based in Central and West African Virus Epidemiology in Ivory Coast. And we have Ntanga Nafoli, who's based in the University of South Africa in South Africa. Professor Apollinaire Jikeng is in the UK. He's a professor of livestock genetics, and he's in the Center of Tropical Livestock Genetics and Health in the UK. Roxana is here, yes. She's the chair of the pilot committee, and we'll get to hear more about it, and is based in the Northwest University in Southern Africa. We have Isidore Wanga, who's our Secretary General. He's also based in the Center for Tropical Livestock Genetics and Health. Thank you very much for providing the links for the meeting. We have Boabid Badoui, who's a coordinator for Northern Africa, based in the Mohammed V University in Morocco. We have Chikraf Salifu, who's based in West Africa, University of Abu Kalbai in Benin. Christian Teambo, who's based in Kenya, but he's a coordinator for Central Africa, based at the Center of Tropical Livestock Genetics and Health in Nairobi. We have Teklu. Teklu is from Addis Ababa University in Ethiopia, but currently in Canada. We have Josiah Kuja, who's in Kenya, based at the Jomo Kenyatta University of Agriculture and Technology. We have Julia, who's the chair of the Genomics for Bioconservation of Endangered Species. And we have Santi, who's the chair of the Bioinformatics and Biogenomics Technologies. We have Aaron Abera, who's the chair of the Technology Development. And Michelle, sorry, I'm never able to pronounce your last name, but he's the current chair of the Socioeconomics, Ethics, and Policy Issues. So that is the steering committee of the African BioGenome Project. Over to you, Thank God.
All right, thank you, Anne. So one of the things we wanted to do, we started to work on this a few months ago, about 18 months ago, but then the initiative started about 2 years ago. So we just went through the first agenda, and we're gonna have to make a presentation on the second agenda to kind of, provide the context to the conversation that we'll be having today. It's gonna be a 90 minutes meeting for today. And of course, there will be time for discussions. And also, apart from that, we'll also be hearing from Eric Jarvis, the chair for the Vertebrate Genome Project. So I will just share my screen. I have the presentation for the next 20 minutes where we'll talk about the African BioGenome Project, and we start, and then I would continue along the way. So can people see my screen?
Not yet.
I'm just gonna share. How about now?
Yes, it's there.
Okay, so I'm gonna just go to the presentation mode. Okay, so maybe Annie, you can just start, and then we'll roll things.
All right, so again, I've introduced myself, and let me just begin by thanking you all for coming and availing yourself to listen to our presentation. This project is known as Africa BioGenome Project. We have a beautiful logo that was developed by an African student. Just in a nutshell, trying to capture what we are trying to do. That is to document through using sequencing the flora and fauna of all various classes present within the African continent. Next. So the next slide just shows you what we're hoping to do, basically to talk about the background, what we're planning to do. And I've introduced you the steering committee. We'll see who the advisors are and also discuss a little bit about what is next. So what are the goals of the African BioGenome Project? The primary goal actually is to coordinate all non-human genome sequencing initiatives that are currently ongoing within Africa, and also to act as a forum for the non-human space. There may be sequencing initiatives that are taking place, but perhaps because they're happening with different platforms, the data can never be merged, so we don't have a comprehensive story. We also want to initiate, develop and support any new non-human genome sequencing initiatives that are coming. We recognize that Africa has very many species of plants and animals, over 105,000, and that is a big challenge, but we're going to start small and build it up within the next 10 years. We also want to deploy all this data to contribute to regional biobanks. So we are aware that sequencing data sometimes is not stored in in Africa, but this is our plan is actually to store our data here and use this data to inform policy and to build capacity within Africa. Thank you. I would like to hand over to Thangod to take us through to the next section, and then I'll come back to wind up.
All right, thank you, Anne. So over the next few slides, I'm going to talk about the development of the Africa BioGenome Project, and then we'll then open up the floor for that discussion and contribution, basically. So as Anne mentioned, the idea basically, or the aim, is to actually sequence genomes of plants, animals, and also eukaryotic microbes across the continent of Africa. So just a bit of a background, putting this into perspective, or having some context to this. So globally, nearly all continents is endowed with a form of biodiversity. And of course, there is also the current event, which is the 6th mass extinction event. And since the 1970s, we've lost up to 52% of all Earth's bird, mammals, and fishes. And of course, in that percentage, we have about 39% of all land species, 39% of all marine, and also 76% of all freshwater species, according to the World Wildlife Fund. Of course, in nearly all of the continents, you also see some form of biodiversity, basically from Africa to North America, and also to Australia. So we do know that Africa plays a significant role in biodiversity, and also there is also an essential component of global biodiversity hotspot, basically. So for instance, this is the map of Africa, and it's got the semi-desert area, the open woodland, and also the evergreen area, mainly nearly all types of forms of biodiversity you would expect. And we also do know that human activities poses significant threat to global biodiversity. And some of these activities mainly involve land use and sea change, pollution, over exploitation, climate change, and also invasive species and diseases, particularly whereby humans tend to interact with the natural environment of these biological organisms. So, but one of the questions now basically is the global biodiversity loss is also happening at an unprecedented scale. So, what this means is that we need an unprecedented effort to actually reverse this trend. And we also believe that Africa must protect and ensure equitable use of biodiversity. For instance, this map shows the biodiversity loss in percentage compared to an intact ecosystem. And as you can see, majority of the areas that have an intact ecosystem have also experienced significant loss in biodiversity. And basically, there are two revolutionary projects that have helped us to advance our knowledge of genomics, or basically life sciences. And one of these projects is the Human Genome Project, and the other, which will of course help us to advance our knowledge, is the Earth BioGenome Project. So the Human Genome Project was completed nearly or exactly 20 years ago. And we do know that it has revolutionized our understanding of human health. And the Earth BioGenome Project was launched about 2-3 years ago. And we do know that it will, in 20 years time, hopefully it will help us to improve our understanding of the biosphere. And the Earth BioGenome Project, which abbreviates for the EBP for short, aims to sequence all 1.5 million eukaryotic species on our planet. And of course, majority of this effort is also targeted at conservation efforts and also agricultural efforts, and just about any other efforts that we could achieve using biological organisms in our ecosystem. So basically, in the one of the things, again, that I started to do when I was a postdoc, mainly is I started to work on what we call the DAISY project, mainly to set up a program or a project where we kind of sequence genomes across Africa. I was working with Rob Davey at that time during my postdoc at the ALM Institute. And of course, Apollonia Jinkeng at the Center for Tropical Livestock Genetics and Health was also involved in that. So when I moved to the EBI, I linked up with Mark Blackster and Harris Lewin to then link up to the Earth BioGenome Project, and the which is now the Africa BioGenome Project. And the AfricaBP is now chaired by Professor Anemugai at the Jomo Kenyatta University in Nairobi, Kenya. So what are the overarching aims of the BioAfrica? One of the things we aim to do is to sequence the genomes of indigenous and endemic biological species across Africa. And of course, the AfricaBP is an umbrella project is an umbrella project for all non-human biological species. That is one of the things we're proposing here. And the aim is to unify all African biogenomes project. And of course, we have the DAISY, which is a community network. Again, I'm going to talk about that in the next few slides, focusing on the agri-environment and also conservation efforts. And the idea is that it's going to be also one of the communities among other communities within the Africa BioGenome Project. And at the moment, we aim to ensure that we're able to generate data, and that, of course, will be used by several agencies and institutions across the continent of Africa, and that will also involve AU, IBA, and also the Africa CDC and other Africa agencies that would actually need this data. And of course, we expect some other projects to connect to this Africa BioGenome Project over the next coming years. So for us, basically, we aim that the DAISY would be an exemplar within the Africa BioGenome Project, mainly focusing on agri-environment and conservation. And DAISY mainly is a community network within AfricaBP to support genome-guided sustainable agri-environment and practices. And the idea, again, as I mentioned previously, is to sequence endemic species, looking at from sequence to applications, also looking at resilient food systems, well-being, and interaction with the environment. And I must mention that there are ongoing activities already, a couple here and there in Africa that aims to sequence Africa genome. For instance, the Africa Orphan Crop Consortium, which aims to sequence over 100 Africa orphan crops, also has been ongoing over the past few years. Again, the idea of JC, the AfricaBP DAISY, is to connect with all of these communities across Africa, and so that we can together sequence these genomes. So for Africa BP, we aim to cover all eukaryotic lineages. And for instance, we aim to look at plants, we aim to look at algae, we aim to look at protozoa, fungi. We also aim to look at vertebrates from mammals to reptiles to amphibians, and of course, invertebrates, arthropod, mollusks, and also nematodes. And over the next few months as well, we're going to be bringing on board scientists, taxonomists that would also work with us on this project. So what do we envisage with Africa BP? The first thing is to build and increase collaborations and capacity, contribute to genomic resources in Africa, inform and drive policy and social economics. And of course, the main aim here is to solve the challenges of the UN Sustainable Development Goals 02, which focuses on zero hunger across the continent of Africa. And of course, this connects to the Africa Union Agenda 2063, which basically aims for an improved utility and preservation of biological resources across the continent of Africa. So, well, one of the things we think at this point is that Africa scientists must contribute to this global biogenome revolution happening around the world, mainly from the Earth Biogenome Project to the Vertebrate Genome Project, to the Giga, to 1000 Genome Project in Chile, to the Darwin Tree of Life, to the European Reference Genome Atlas, and also to the arthropod genomes, particularly. And one of the things, again, that we've been asked over the past few months is it possible to sequence all eukaryotic organisms across Africa? And of course, I've mentioned basically projects that are ongoing around the world in nearly all the continents, focusing on sequencing on either taxa, a particular taxonomic tag, or focusing on a particular community or a particular region, basically. And of course, I've talked about the Sanger Tree of Life, the Darwin Tree of Life, and the VGP. And the Earth Biogenome Project is more of an umbrella community that kind of houses majority of this project, basically across the world. And of course, we also believe that biodiversity genomics in Africa must be driven by African scientists and agencies across the continent of Africa. So there are 5 challenges, grand challenges, mainly that we aim to solve within the Africa BP. And one of this is genomics and bioinformatics technologies for the agri-environment. The second is crops and livestock improvement and health. The third is genomics for conservation of endangered species. And the fourth is socioeconomics and policy. And the fifth is technology development, knowledge exchange, and industry. And of course, all of this revolves around value proposition and capacity building across the continent of Africa. So for us in the Africa BP, we aim to drive standards across the continent on biogenomes activities. And of course, would follow this typical standard of biogenomics, mainly involving sampling, nucleic acid preparation and extraction, sequencing, informatics, and of course, value proposition and empowerment of the communities. So looking ahead, initially, we were going to launch this project at the Biodiversity Genomics Conference, which is hosted from the Sanger Institute so in this coming September. But one of the things we are trying to do is to move this until sometime in 2022 or 2023, mainly because we aim that this project should sit within the Africa Academy of Sciences or any other agencies in Africa to be able to better target the people and deliver value to the people. And over the past few months and continuously as well, over the next few months as well, we will be engaging with scientists, policymakers across the continents and other people who will be interested in this project. So at the moment, we are aiming to commence a pilot project. Again, one of the things we wanted to do was to commence pilot next year, but then there's also an immediate opportunity for us to collaborate to commence this pilot in 2021. So this month we'll commence the pilot project. We've actually sent out call for that in terms of species, specific species. And I'm going to talk about this over the next coming slide. So after the pilot in 2024, it's going to last for 3 years. We'll progress on to have the phase 1 and the phase 2 project. So we expect that this will be a multi-step process over the next 10 to 15 years for Africa BP. So we also aim to employ a strategic partnership framework. It's going to involve a multinational partnership, multi-country partnership, mainly, and also involving multiple organizations and institutes. Particularly, we aim to work with the AU-IBA, Africa Academy of Sciences, and hopefully the Africa CDC, looking at data coming from biodiversity, particularly, and also work with History Bionet, which have also helped to build the genomics capacity across Africa, and so many other partners across the continent and outside the continent that we could actually see their logo here. So as I mentioned previously, we aim to commence a pilot project on AfricaBP. And the idea is to focus on about 2,500 species in 3 years. And this project, basically, we aim to work with the Vertebrate Genome Project. So Eric will be talking about this, the VGP project in the next presentation, basically. And the idea is to work with the VGP to sequence about 500 vertebrate species across the continent of Africa. And of course, we'll be working with the Africa Academy of Sciences to deliver on this. And the idea, again, is to onboard this onto the ESA program in the AAS. And again, we're open to partnerships to deliver other eukaryotic taxa within the pilot project. So we're not focusing on just vertebrates, we're also looking at other taxa within the pilot project. So I'm going to hand over to Anne again to talk through some of the things on how she aim to run this across the continent. Anne, over to you, please.
Thank you very much. So when we started this project, I think we were a bit overwhelmed because of the number of species and the diversity of our terrain and thinking about where we're going to start. And when you think we have about 2,500 native and endemic species, and among that we have the endangered species, you tend to see what the challenges are. However, we want to work on representatives from all taxa, so animals, plants, fungi, seaweeds, macroalgae, and we're going to draw on the strengths and the passions of the scientists that are in the continent. Of course, the focus would be on endangered species and those species that are of economic use to Africa. Next. So we will engage scientists who are naturally working in Kenya, in Africa, and approved custodians of the samples. And this will be coordinated by the pilot committee. Any fresh material that is going to be needed will be collected in collaboration with the scientists who are present in those countries, including national, private parks, nature conservation centers, and other custodians of this material. We are also looking for taxonomists who will help us identify and classify this material as we are collecting them. And of course, we cannot forget that there's material that is already being stored within Africa in biobanks. And this is also a target, and we'll see how we'll be able to use this material for our project. Next. So we are also looking at other partnerships that can be able to work closely together. For instance, the EGRA committee, the European Reference and Genome Atlas, are very, very interested in some of the cross-boundary species. So any initiative and any collaboration that will help us work and sequence the African biogenome, we are very, very much interested in it. Thank you. Next. So we have to consider some of the capacities that are built across Africa. And when you consider the many institutions that are within Africa that have capacity to carry out some of this work, case in point, the Biosciences East and Central Africa, the IITA, the H3 Africa, and therefore we're going to work collaboratively to be able to build capacity and also to be able to carry out this work within the continent. Next. One of the things that we had to look at is where does Africa stand when we are looking at the genomic capacities? And following data that we found published, there are 206 genomic capacities in the continent. 71% of these are concentrated in 5 countries. 70% of these are outside National Public Health Institute. But for us, we are going to do the sequencing, data collection, sample collection, all the work will be done within the continent. So it's important for us to have one platform where this work can be carried out. And of course, this will be in partnership with these laboratories that have this capacity. Next. One of the things that we have to consider is how we can collect samples considering the Nagoya protocol. Of course, within Africa, you know that right now, we have no clear attribution of responsibility on the enforcement of the access and benefit sharing. And we also don't have an effective mechanism on how we can bring in partnership and collaborators. The regime is unclear in terms of regulatory, but we are going to navigate this collectively as Africa, and we're going to have discussions on how we can be able to collect samples from different countries. And if there's no sequencing capacity within that country to see how we can move them to centralized laboratories within the continent. And this data will be available for all the member states to share. Next. We are also conscious of the challenges within the terrain in terms of the landscape. It's very, very wide. The samples could be in very remote areas. And therefore, the challenge of actually accessing these samples is something that we have come to consider. And we realize that the model that we may have to use is have more of portable molecular biology interfaces or laboratories where we can travel with compact, portable equipment that is inclusive, and we put them all in a box and travel to a particular location, sample them and collect samples and sequence, and using Wi-Fi start uploading sequence information. Of course, this presents challenges because probably this equipment has to be made smaller and portable, and has to consider the fact that we don't have electricity connection in all parts of Africa. But this is something that we'll have to see how we can work around. Next. We are aware that one of the greatest costs in Africa is actually the access to internet. And we also are aware that the internet is not available in all parts of Africa, and it's not stable. We are aware that when you consider that, and you can see from this publication, that when you consider the amount of cost Africans are paying for access to information and Wi-Fi, it is much, much more than what is being paid for in other areas like India, for instance. So this is something that we're aware of, and we'll have to navigate this challenge because the genomics platforms have a need for internet access, which is fast, which is efficient, and we'll have to navigate how we'll go about this. Next. I want to present for you the organogram that we have come up with for the African BioGenome Project. At the top there, you'll see the steering committees, and then you'll have the Science and Technology Committee at the second level. But I'd like you to focus on the fact that we have nodes, and each part of Africa has a node which is headed by a coordinator. These particular nodes will be the ones to identify the samples, location, shortlist the samples that will be sequenced first, and actually coordinate how these samples will be transported to laboratories, whether they're in the country or in the next neighboring country. And we hope that this will ensure that we have collective voices from the entire continent, so that we move together without any part of Africa being left behind. And it's going to be an inclusive effort. And I hope that you can participate in the discussion on which samples you would like to have first, and then we can collectively make an agreement on how to move forward. Next. I've already introduced you to the members of the steering committee. And next, in support also, we have, yes, these are the names of the people. We have an advisory team. We recognize that we will need to have advisors who will ensure that we remain on track and offer the expertise. And we have Nicola from the H3 Bionet. We have Rob David from the Elam Institute, and Simplis Nwala from the African Union based in Ethiopia. Thank God, you can continue.
All right, thank you, Anne. So I'm just gonna move a bit quickly because we have Erich who will be talking to us, and I also understand he has another meeting after the presentation. So one of the things basically we've looked at is, for instance, what are the possible funding mechanisms for AfricaBP? I mentioned that basically we're looking at least about 100,000 species. And of course, based on a rough estimates, this will cost about approximately $1 billion. And in terms of sequencing and computing costs, and that does not take into account the actual logistics that will be required. And this funding, again, is looking at it in about 10 years. So we do not expect this funding to come from one particular source. So one of the things we are looking at is having a supplementary funding, mainly leveraging on the global communities within the AfricaBP Biogenomes Project, such as the Vertebrate Genome Project, who have existing project to sequence species across the world. So again, we'll look at all the species that are endemic to the continent of Africa. And from early 2022, we'll be working with the VGP in the phase 1 and phase 2 project to sequence about 500 species across the continent of Africa. And again, the supplementary funding too, we aim to leverage on existing projects. For instance, the African orphan crops, we'll basically work with them in order to deliver some of these projects, and also other projects across the continent of Africa. And of course, we also aim to work with the Africa Academy of Sciences, because one of the things is that we feel that this project should actually sit within the AAS or any other strong agency across the continent of Africa. So we aim to work with them in order to be able to get core funding for the delivery of the main objective for this project. So, but one might ask, should Africa commit $1 billion to biodiversity genomics in the face of poverty? Can we actually directly feed the population with this money, basically? But I and also many other basically think that this presents a short-term solution, and we also believe that to build capacity in order to actually produce the food for the future is actually a long-term goal. And of course, that would basically understand that there could be argument in terms of looking at what should we or should we not do in the face of this particular challenge that we have. But then there are basically several reasons why we think we should do this. For instance, the Human Genome Project, which again I mentioned previously, costed about $3.3 billion. But just in 2019 alone, basically it generated revenue of $108 billion. And we strongly believe that the impact of the EBP will be far-reaching in our biosphere, meaning looking at plants, animals, and other systems. And again, within the context of the Human Genome Project, we do know that at least up to $3.3 billion basically was basically committed to this. And again, it's generated over 150,000 jobs, supported 850,000 jobs again, and have an overall direct federal tax of about 5.2 billion revenue, basically. So we could see the benefit of some of this. But then again, within the context of Africa, we might ask, why must Africa act now? Why not tomorrow? Why not in 5 years' time? Why must we invest basically in this huge ambitious project? So the reasons basically mainly involve that as the EBP gains momentum, Africa must avoid the mistakes of the Human Genome Project. So again, I mentioned previously that the Human Genome Project and the Earth BioGenome Project are two projects when it comes to life sciences, that one of them, which is the Human Genome Project, has helped us to advance our knowledge. And we believe that in 20 years' time, we'll begin to see the benefit of the EBP. Well, one major challenge of the HGP is that it's missed important gene variants in the African people, mainly because there was little or minimal participation of the African people in the HGP. And what that meant is that we also have about 300 million letters of the DNA of the human genome missing, particularly. This also despite the fact that Africa has the higher genetic diversity. So we believe that the participation of Africa in this will also be very helpful, because it will help Africa build capacity, and particularly when it comes to combating pandemics and several other challenges across the continent. So again, we can see how African scientists help to improve cassava in order to feed the world, and also in terms of conservation. So what is the charge to African scientists in the area of the biodiversity genomics? So one of the things we believe is that we should be able to look back at this era with hope and confidence. But in order for us to do that, we must be able to act. And we also believe that African scientists must lead the way in sequencing African biogenomes, basically in this era. And one of the things we're thinking about is that, of course, by the time we finish the sequencing all life on Earth, that we could basically have several publications and several things to show for in terms of Africa's contribution to this project. And in order for the AfricaBP to be successful, African scientists must galvanize efforts at the national level. So what is next for us, basically taking off and moving on? What do we hope to do, basically? Over the past several months, one of the things we've been doing is consultation, engaging scientists, engaging policymakers. Should we do this? Is this a good idea? And also engaging other people and other stakeholders in this. And at this point, we feel that it's now time to engage the general public, basically. So we started to write a position paper, which at the moment we have a draft of the paper. So we aim to publish that, and then we have a white paper which is also under development. And we aim to commence the pilot studies with the VGP and populate a list of 2,000 species, and of course work with the African Academy of Sciences to onboard this program onto the AAS. So we have a temporary website which you can just at the moment connect, kind of visualize things. And also we have a Twitter account Daisy_AfricaBP that you can follow us on. And there are up to 350 email subscribers to the AfricaBP at the moment. So, and this is the Google Groups. You can just send an email there and then you can subscribe to it. With this, I would like to thank every one of us for listening. And I would maybe stop sharing my screen and we can then move on to, so the idea is that we want to be able to merge all the discussion together, that of Erich and also that of the AfricaBP. So maybe we can listen to Erich talk over the next few minutes and then we'll merge all the conversation and Q&A session together. Over to you, Erich. I think you're on mute.
Yes, I have to unmute. Okay, well, thank you for inviting me to give a presentation here as well as to contribute and as well as to explore and share ideas and collaborate. So, so thank God asked me to present some of the lessons we've learned in the Vertebrate Genomes Project. And from those lessons we've learned, recommendations I can make for the AfricaBP project. And so, now you said about 20-minute presentation. I'll try to limit it to that. We'll see. I have a bunch of slides here. So, can you see my screen?
Yes, we can.
Yes.
Okay, fantastic.
We can.
All right, so I titled this not just only the Vertebrate Genomes Project, but underlying mission of this project has been sequencing high quality genomes, which I will call, I think will bring in a new age of biology, and you'll see why it makes a difference in investing in high quality genome data as opposed to draft quality. And so the mission of the Vertebrate Genomes Project since about 2015 has been to generate at least one high quality, error-free, near gapless chromosomal level and the haplotype phase for diploid or multiploid genomes, annotated reference genome for all extant vertebrate species, and to utilize those genomes to address fundamental questions in biology, disease, and conservation. So the mission is both genome assembly based in terms of quality, as well as scientific based in terms of generating genomes that can address particular questions. And for example, in these three main areas, biology, disease, and conservation, and other areas as well. And when we began, we took a more phylogenetic approach compared to some of the other projects that are happening now. And this was born out of the out of the Genome 10K initiative, which started back in 2009. And for going for all vertebrates, our goal was to first focus on all orders, then families, then genera, and then eventually 71,000 species. And this includes efforts globally. Edith, if you can mute. So, and each one of these, stages, we saw as other projects started to develop, would reach certain milestones. For example, phase 2 would be all. Okay, can you hear me still?
We can hear you now. Go ahead, please.
Yeah, great. Okay, good. All right, so phase 2, assuming we complete the VGP phase 2, that would represent all Earth BioGenome Project families. Phase 3 would be the original G10K milestone of 10,000 species, in this case representing all genera. And then phase 3 would represent all species of vertebrates, at least for all the projects that are interested in vertebrates. And so since that period of time, we formed in as multiple groups within the VGP G10K organization, like a council, samples collection group, and so forth. And I would say that the group that turned out to, I would say, make a huge difference to the project overall is the assembly working group, which I would encourage folks from the AfricaBP group to become part of. This is chaired by Adam Philippi from NIH, who's one of the leading assembly experts in the scientific community, but many of you will recognize many names here, including those from Sanger and others. And this group has grown since this image was put together. And it's now a combined VGP-EVP assembly group. And so There's a more recent work we just published that some of you may know about in a special issue in Nature, which they titled "The Spinal Frontier," meaning the spinal cords of vertebrates, where we started out with a hummingbird to test a lot of this technology. And there's a series of papers on the genome technology as well as on the biological discoveries from 16 recently assembled genomes using these high quality approaches. And I encourage you to go look at that. I'm going to point out a few of the main messages here. Okay. One coming from my own work, I'm not necessarily a genome biologist in terms of my initial training. I'm a neuroscientist studying brain pathways for vocal learning, and certain species have that ability, including parrots that are in Africa, songbirds around the world, hummingbirds as well. And there are certain brain regions that we found have convergent changes in gene regulation. White is the mRNA signal here, the messenger RNA in these brain regions are the 3 vocal learning bird groups compared to non-vocal learners like doves, like pigeons and quails. And we find that they have convergent down regulation, turning off of this gene that's involved in connections in the brain. And so do they in human speech areas, which is called the dorsal and ventral laryngeal cortex. And what got me involved more heavily in the VGP and other efforts is many students in my lab, we found hundreds of genes that were convergent in the brains of songbirds and humans and parrots. And we wanted to study these genes. And in the draft assemblies generated with the initial Sanger technology and also Illumina short read technologies, we were finding that these genes were interested and were highly fragmented. Here is a zebrafinch gene, the SLIT1 gene, 35 exons spread over multiple contigs with lots of gaps that were in coding and non-coding regions. And when we sequence genomes with short reads of many species, we found that we're missing many genes or parts of genes. Many genes were incorrectly assembled. Biological meaningful repetitive regions were misassembled or not assembled at all. This is causing many months per gene to clone and correct gene structure and sequence for members of my lab in my collaborations. And we are unknowingly working with artifactual gene structure and sequence, unknowingly making false conclusions about the biology. And that lesson taught me I can't do it. It's a moral lesson, and for scientific accuracy, and also to answer the questions we needed to answer. So what we ended up doing at the start of the Vertebrate Genomes Initiative for high quality genomes, we took one of these vocal learning species, a hummingbird, and at that time applied all the available technologies to it. And each of the sequencing companies that were involved, most of them actually did it for us for free because they wanted us to utilize their technology for thousands of genomes going forward. And we set a minimum metric that allowed us to generate novel biological discoveries with as little error as possible. And that minimum metric was the contigs have to be greater than 1 million base pairs. The scaffolds have to be bigger than an NG50 of 10 million base pairs. Assigned the entire assembled scaffolds to at least 90% of them to chromosomes. Have a base call accuracy that's the AGCT accuracy greater than 40, which is mean no less than 1 error every 10,000 base pairs. And we found that we actually have to phase the haplotypes because when we do that, we actually get less errors, and you'll see in a minute. And we came up with a pipeline that began with long reads, in this case, PacBio continuous long reads at that time, and to create the biggest contigs we can, then scaffold, in this case, with 3 different technologies, 10x linked reads, bionano optical maps, and high-C chromosome interaction maps, and then polish to get the base calls accurate, then curate. We noticed that even today, we still need to curate to fix errors. Even with the best automated assembly tools, if you don't curate, you're going to have a bunch of errors that you don't want. And so, so we applied an approach that we found worked well in the hummingbird to 16 different species, going from long reads in the red here to long range scaffolding data in the purple here for high C. And this was a collaboration of many people. It wasn't just one individual lab doing this. This was a collaboration across labs, across countries. And this is just one graph to show you the difference long reads make. And I can't show this enough because there are still folks out there who believe that the short reads are really going to generate the quality we need, and it's not. So here are the contigs, and the contigs are hundreds of fold orders of magnitude bigger than contigs with short reads. Here are the scaffolds. It doesn't make too much of a difference with long reads on the top here and short reads on the bottom here to make scaffolds. But when you go and look at the structure of those scaffolds generated with short reads, you have about 70,000, 18,000 to 70,000 gaps compared to several hundred in the short read based assemblies. And you have lots of misjoin errors, like 5,000 to 7,000 misjoin errors of contigs in these short read based assemblies, wherein they're in the hundreds, which is more easily fixable, for the long read based assemblies. What's nice, and as many of you know with these high C plots, you can actually generate what, after curation, like the Kerson's Howe team at Sanger, they curated these genomes, get pretty much almost arm to arm chromosome assemblies with some gaps and so forth that match in size nearly 1-to-1 the karyotypes of these genomes. Work, again, done by collaborations with various different groups. Here from Joanne Adamus from Harris-Lewin's group, compared the old assemblies based upon the Sanger and the Illumina technologies with these new long read assemblies, and anywhere where there's white, that wasn't sequenced before. For example, in the zebrafinch, we identified 8 new chromosomes that wasn't identified before, and these turned out to be GC-rich chromosomes. They'll come up in a minute. We found that previous assemblies that split chromosomes apart thought there were 2 separate chromosomes, are really 1. We also found here in the platypus, was it 8? That was the case. There were many splits in what was annotated as 2 different chromosomes of 1 because we're bridging gaps and bringing them together. Also newly identified, about 10 newly identified chromosomes in the platypus, and identified also the sex chromosomes, 10 sex chromosomes altogether, 5 X's and 5 Y's. And so what's going on? What we see are missing in these prior assemblies that were based upon the short reads only. And a predominant thing that we're seeing that's missing, regardless of species, is red here. So this is a plot of the zebrafinch, the hummingbird, the platypus, a fish here. And from the chromosome sizes or scaffold sizes from the biggest to the smallest, the log scale here. And everything in red was missing in assemblies based upon short reads or Sanger reads. And basically what we're seeing is that these are repetitive or GC-rich chromosomes. Okay, and so all the purple here in the circus plots are microchromosomes that were missing in the prior zebrafinch assembly that contained roughly about 1,000 genes that were once thought lost in birds. All right, and it's not just anywhere in the chromosomes that you have missing sequence. It's particularly there's an enrichment of missing sequence in the promoter regions. So what we see here in red of various different vertebrate lineages or species is the percent missing sequence as we get closer to the 5' UTR. So this is roughly 2,000 base pairs upstream of the coding sequence. And we see as we get closer and closer to the 5' started gene, we have more and more missing sequence in the short read and the Sanger-based assemblies. And so when we look at the GC content in the new assemblies, we find correlated with that is an increased GC content as you get closer to this 5' UTR. And the 5' UTR and the exons of all these genes in these new assemblies are also GC rich. The introns are not. The introns are the same as the general genome background. And you can see that pattern here, just, now the 16 genomes graphed together now, mammals and birds have the highest GC content right in front of their promoter regions, followed by the reptiles and amphibians, followed by the fishes, the least. And so what we found is that in the old assemblies, not even knowing it, we're missing anywhere from 26% to 60% of the genes are missing some sequence. Mostly GC-rich regions, but also gaps in the assemblies, errors in the actual sequences itself, and so on. So that's a lot of data that's missing. Going back to the SLIT1 gene I told you about before, we now have it completely assembled in all of our bird and mammal genomes. And we found that the missing sequence in the older assemblies, what happens when Ensemble and NCBI annotate that, there's a missing exon here. So they take the methionine of the next one upstream, which is part of the promoter region, and incorrectly annotate it as a protein coding sequence that does not exist in nature. in these regions that have gaps in the assemblies. And it's a GC-rich region. Another thing, the haplotype phasing I told you about, the lesson we learned here is this is a gene that's upregulated in the songbird brain regions for imitation of when the birds are singing. So the white signal again is the mRNA. And we find this gene is not regulated in the surrounding motor areas when the birds are doing other things like hopping. And we found that all vocal learners, we think what's causing this specialized upregulation in the song-like or speech-like areas is a repetitive region in the promoter. But what we found is that that repetitive region was incorrectly assembled in the old assemblies, whether it's short reads or even incorrectly assembled in the long reads, unless we phase the haplotypes into alternative haplotypes. We found that these repeat regions in both haplotypes were so divergent from each other that the initial assemblers thought they were two separate, sequences, where they're the alternative haplotype. So if you don't phase, you also get lots of errors. You can even get the entire gene is divergent between the two haplotypes, and the assemblers will make a gene copy A and a gene copy B. They're really not paralogs, they're really homologs on the two haplotypes. And you can get exons that are being inserted from one gene into another as well as duplications. And we found that we're also expanding the sizes of many genes, like this L and CAM1 or oxytocin receptors, because so many gaps were in them before, we didn't really know the true gene sizes, but now you fill them in, you actually realize they're bigger. And this whole region is now assembled on one conte. But it's not all rosy. There's still remaining problems. One problem is the more repeat structure you have, the more they get collapsed, particularly in centromeres and telomeres, but also duplicated genes. Even though we purge haplotypes from the assembly, you can get assemblies that are, purged false duplications. You can still get assemblies that are bigger than the estimated genome size because there are false duplications we're still not getting rid of. So we do have some problems still to fix going forward in the future, even though these assemblies are good. But they're good to the point, especially when you manually curate, like in this hummingbird chromosome 14, it's nearly telomere to telomere just with one gap. The bird genomes are easier to assemble than the mammal genomes. And efforts that learned lessons from the VGP and made further advancements in the Human Telomere Consortium just submitted something to bioRxiv, where through a lot of manual curation with these long read approaches and scaffolding approaches, we're able to get the first human telomere to telomere genome. Now, this is mostly a haploid cell line, so it makes the separation of haplotypes much more simple, and so you have less errors that way. We still don't have a diploid mammalian telomere to telomere assembly. So future plans for the VGP are to complete phase 1, which is 260 ordinal species representing all vertebrate orders, conduct foundational studies on these vertebrate orders like the vertebrate family tree, for example, at the ordinal level. A version 2 approach for the assembly going forward for these at least 260 species. We're combining high 5 with high C and bi-nano. So we've been able to eliminate a lot of scaffolding data because the new long read data is more accurate than we were using before. We haven't yet set annotation and alignment standards. That still needs to be done. And I think that's going to be happening in the next phase of this project. Funding for the next phase is really, we're going to need to, what Thank God talked about, 1,200 vertebrate families, which I estimate there are about 300 to 500 of these vertebrate families or species representing them in Africa alone. And the VGP and EBP assembly group is now going to start to work on approaches to try to generate T-to-T diploid assemblies of non-human vertebrate species, but also we're thinking about bringing in others for plants and insects. And so eventually, I think we need to go as a consortium. All these consortiums need to be at a stage where you generate a genome once. If you're going to do all species on the planet, all species in Africa, you don't want to spend all that money and do it multiple times. So I think we need to get to a place where we just do it once. And so, so these teams of people coming together are not just generating the genomes for biological investigations, but also helping to advance the technology. And here for these 260 species of vertebrate orders, some of them are already African species that we have done. And so why 50 million years? Because it goes back to the time of the last mass extinction of dinosaurs, or the last mass extinction period. And so we can understand from doing all orders with a 50 million year divergence time, what happened during that last mass extinction to inform our current mass extinction. And so, and what I see also going forward is that there have been three major kinds of projects that have been developing. A phylogenetic type of project like the VGP or GIGA or EBP, but the geographic or nationalistic projects like Darwin Tree of Life, European Reference Genome Atlas, Africa EBP, and the Madagascar one also that some of you may know about. And the pan-species one, there's a new one, Human Pan-Reference Genome, where you produce high-quality genomes within a species representing the species diversity. And each one of them have their individual organizations and websites now. And what I think the EBP is really set to do is really bring all of these groups together. And the three biggest lessons we've learned going forward in the genome assembly world is haplotypes of one giant repeat. They must be dealt with properly. Read lengths need to be longer than the repeats themselves within the same haplotype to resolve those repeats. Sequencing accuracy, the base calls and the phasings need to be high enough to prevent false gene duplications. And you need sequencing technology that not only gets through repeats, but high GC content to get your promoter regions, to get interesting microchromosomes that are GC rich. This is probably the most important slide for this group, and my last one here is that my recommendations, these are my personal ones, but I think many people will share them with me, is that where I think Africa BP contribute to not only Africa's BP's project, but to the world at large, right, is really put a lot of effort into sample collection and coordination. This is a weak point in many of these projects that are global. And so I'm working on developing permits with individual countries or the continent that are more, that are blanket-like permits, inclusive for collecting and receiving and sending samples within countries and across countries. And I would recommend joining the VGP EBP Friday assembly calls, having genome experts join those calls and be part of that team, because making a separate one or having a separate one is okay, but I think at least being part of this one where you have some of the world leading genome biologists in this group every day on every Friday, which will be on the afternoon or evening times in African countries, contributing and learning from this group is, I think, is really going to make a difference for all these projects. Data production groups, like Thank God said, I think beginning with some of the existing hubs, like for the VGP or the Darwin Tree of Life, mostly now focused in Europe and in the United States, to do some genomes for the AfricaBP, but eventually, like we've heard here, setting up hubs in Africa would also be important. And setting up those hubs, once you set them up, they're going to be expensive right now. They're in the millions of dollars to set up a hub to produce high quality genomes in terms of hiring people, buying these sequencing equipment and so forth. But you need to be ready to upgrade every year. It's not once you spend the $1 to $2 million, you set the hub up. Once it's set up or you upgrade an already existing hub, until we get to telomere to telomeres, this technology is going to keep continuing to develop every year. Assembly and curation, I think, again, coordinating with the existing efforts to train curators within Africa to take these genomes to fix the errors, all right, that you can fix manually. Annotations to make sure that samples not only collected like blood samples for the genome, but if possible to collect at least 6 or more tissues to create transcriptome data that's necessary for annotating the genomes. And annotation, I think, should be species-specific annotations. Setting up website and databases that integrate with each other, not just standalone websites. Like we have something called the Genome Arc, where it's like a working website where we put all the long read, short read, and other data in there to create the assemblies before we pass it on to Ensemble and NCBI. And annual meetings, I think being part of this biogenomics diversity meeting, genomics, you know what I mean, which began actually as our previous G10K VGP meeting. And so we don't have a separate VGP meeting anymore. It's part of this group. Outreach and funding, I think this is also a big area where Africa BP could actually make a big dent into these global projects, is having a lot of outreach. So I will say also, I think scientists within Africa can benefit from these genomes and generate novel biological discoveries. where I think we're still having a weak point in these global projects. So it's already a big collaboration. These are authors on that flagship paper that came out on the VGP. And I want to thank you for listening. And I'll stop there and just show, website here that we have the deposition of all these different genomes for the Vertebrate Genomes Project, for high quality genomes here, all these species listed, that links to the Earth BioGenome Project as well, page for a public database. And I think AfricaBP then should have a link in here as well, which will have the African genomes that are listed under the EVP here. And now I'll stop.
Thank you, Erich. That was a very nice presentation. So I understand you have another meeting at 4. Is that still going on, or?
No, I can stay another 20 minutes.
Okay, all right, perfect. So
if you have questions, go right ahead and answer them, or suggestions.
Yeah, so I will just open the floor to people so to kind of comment, make suggestions, ask questions. So I think that because there are about over 120 people on the call at the moment, if you could please maybe post your questions on the chat box or raise your hands, then we could just open up, maybe select people to ask questions. So over the next 30 minutes, we're going to,
okay.
over the next 20 minutes, we're going to respond to questions, get contributions, and then make some clarifications that are required. Okay.
Yeah,
so,
and so I see people raising their hands up as well, like
okay.
Harry.
All right. So I think Harish, could you please unmute and ask your question? And please try and mute if you're not speaking, so others could benefit from the question. Go ahead, Harish.
I was going through your paper on the bioarchive as well as on the nature set. So I just wanted to have this question with you as to, you recommend having like 6 different tissues for transcriptome sequencing. And when you're speaking about transcriptomes, are you also including isoseq? Or are you just talking about the standard Illumina-based sequencing? Because I'm wondering if just having 6 tissues, or say even for samples which are like highly endangered, getting 6 tissues would be a lot difficult, especially in the quantities that is required for isoseq. So how do you propose that we mitigate this issue?
Yeah, this is
kind of my question. Thank you.
Okay, good question. This is an ongoing question even for the assembly group and the annotation team. And there's still debate about you're getting good coverage across genes when you do the Illumina RNA-seq method. But you're getting more complete and accurate gene annotation structure when you're using the isoseq or the nanopore long reads. I think we're going to be moving more and more to the hi-fi isoseq reads because it's more accurate now. And there's no assembly required, it's just a read through an RNA transcript. And the coverage is now higher than it used to be. So that's my prediction going forward for the coming year. 6 tissues, what the ensemble and NCBI teams have found, at least with vertebrate species, when you have 6 diverse tissues like brain and liver and muscle and so forth, you start to plateau at the number of newly discovered transcripts at about 6. But annotation is a bottomless pot because genes are being turned off at different developmental windows in different individuals, different populations will express different genes and so forth. So it's going to it's impossible to even annotate the entire human genome now. So you just want to get an annotation that I would say that's roughly going to be 80% or more of the genes. And I think 6 tissues cover them.
All right, thank you, Eric, for the response. So I'm just going to go to the chat box now. There are about a couple of more questions there, so we're going to attend to that and then come back to those that are raising their hands up. So Yonahan Tilahun asks, is there a consideration for an African biome project? So I think I'm just going to take that on. One of the things we aim to do, basically, again, we've discussed this with Apollinia and several other members of the steering committee, is to focus mainly on the eukaryotic side of things. And yes, in the future, we aim to expand into that aspect, but in the interim and for now and going forward, we want to focus on the eukaryotic biological system. And of course, we could have an Africa biome project, basically that tend to connect into the overall Africa BioGenome Project. Again, just as I mentioned, the idea is to make this an umbrella project, and then we could have other sub-projects that connect to it. I hope that answered your question. Yes, I think you asked if we can integrate it with BioGenome. Yes, of course. Then Martin asked, have center.
Thank God. Can I add quickly to that microbiome one?
Okay.
we're finding with these long read sequencing technologies that in the samples that we sequence, not only do we sequence through an entire mitochondrial genome, but we sequence some of the bacterial genomes that are inside the sample, meaning the microbiome of the individual at the time. And normally we just throw them away. They're sequenced, we just throw them away and say this is contamination. But I think in the future we should really be thinking about saving those sequences as part of the assembly, because these are species living in the individual animal or human.
Thank you, Eric. Then I think I would take one more of this in the chat box, and then we'll go back to those raising their hands up. So someone asked if we should, do we have central sites? Have central sites been identified as part of the main hubs and protocol standardized? So I hope I can take that as well, basically. So one of the things we're doing, again, as I mentioned, is over the past 18 months, 2 years, is to speak to people whether we should do this and then look at what we should do, basically, and mainly kind of building support. So at the moment, we've set up the pilot committee, and within that committee, we have about 8 subcommittees made up of about 10 to 15 people. So one of the things we're giving to the sample collection and processing committee is to be able to identify some of the standard protocols and look at which site, what do we need to do particularly. And as Arish mentioned, basically one of the practice is going towards having the hi-fi technology. Again, last week we did have a meeting with the South Africa, Nkaba and the PacBio leadership, mainly to begin to look at some of these things. But I think the best thing would be at the moment to start building the capacity from where we have them, rather than trying to build everything at once. So I hope that answers your question, Martin. And of course, we also aim to have both the mobile sequencing and as well as reference laboratories or center site. So Uche, you do have a question, could you please?
Okay, thank you, Angod and the team for the wonderful project. So I think you mentioned about the funding. This is a very big project that will require a lot of money, though you mentioned about $1 billion going to be used for this project. So I want to know, like, have you really already secured this funding, or are you still? So what is the progress so far in that aspect? Then also about the database, I don't think you mentioned whether this project will have their own database, because I saw where you are talking about different databases that you already have. You are going to store the data. So you are in this project now, there is no your own database. You're not going to make your own database. And finally, we currently have about 54 African countries. And you mentioned that in the first phase, you are going to sequence about 2,500 species. So in this case, how are you going to select it across the continent? Are you going to make sure that each of these species come from at least one of these African countries? So thank you.
Yeah, thank you, Uche. So the first one that has to do with funding, we haven't secured the funding yet. And just as I mentioned, the conversation is there, basically. And our aim at the moment is to focus on building the collaboration needed for the supplementary funding 1 and 2. And we're also in conversation with the African Academy of Sciences, particularly. But one good thing about this project is that it's the kind of support we've gotten both from the continent and from outside the continent. And also in terms of having some species, trying to sequence some vertebrate species. And in the next few weeks as well, I'll also be having a meeting with an investigator in Cambridge, mainly looking at plant species as well and several other communities. So we haven't secured the funding yet, but the idea is to begin the analysis with the Vertebrate Genome Project, and they will basically leverage on the existing funding for the number of species that we aim to sequence with the VGP, and while we begin to make inroads into the other communities or taxa. And ultimately, we would aim that this project will sit within the African Academy of Sciences. So the second one has to do with database. So I understand that this is also a very important one, and it's a question that's something that we've discussed again with Apollinia. Maybe I'll also allow him to speak just very briefly. is that we're generating all of this data. Yeah, of course, we can store them in public databases and, in the US and Europe. So there are also other interests that, okay, should the project actually have its own database? I think, again, it requires huge amount of funding and commitment and financial implications. So that is a question or something that the African people have to think about, and regional governments. So I'm going to give Apollonia maybe a few minutes to talk about that bit. But your last question.
Sorry, go ahead.
Yeah, your last question, Uche, you mentioned about, could you please remind me again, the last one?
The last one is about the number of African countries we have, okay, and how you are going to represent these species.
Yeah, so one of the things we're trying to do, again, I mentioned we set up the pilot, is that at least we would have representative species from each African country. And just as Anne mentioned, we want to be very inclusive. We don't want to leave any African country behind. It's going to be Pan-African. So even in this pilot project, we're starting with 18 species. We can't distribute all of the 18 across the 54 nations. So what we plan to do is that each country, each region in Africa will at least have one species represented. So Apollonia, I don't know, do you want to make some contribution? Apollonia is a co-chair in AfricaBP. Basically, with regards to the data side of things, well, I think this is a very important aspect for the project. And I know we've had that conversation multiple times.
Yeah, thanks, thank God. And thanks also, Rich, for the presentation. So this is just starting, but we are starting this considering some key principles: data ownership, origin, and retaining things in Africa as much as possible to build capacity and have sequencing in Africa is really, really important. But there is another aspect, which is the access benefit sharing. The Nagoya Protocol is going to be another very important consideration as we move forward. And you can imagine, Uche, that from the very beginning, it would be hard to have every country with a sample, but the sample committee is going to consider samples that belong to various countries or to just a unique country and stuff like that. So these are the guiding principles that we want to use moving forward, but as much as possible, the database, the data repository and things like that will be available and accessible to scientists in Africa, because the ultimate goal here is not just to sequence things, it's really to transform the way we do research in Africa and get some of the labs, considering this as starting materials or starting resources for work to happen in their labs and things like that. I also want to touch on the component on the microbiome. We've not neglected that. We just want to make sure that we are focused. The microbiome component will be very, very important for us to understand other dimensions for the organisms that we are sequencing, but also more importantly on the environmental part of it. So it's not neglected, but we cannot really pick up everything at the beginning. In terms of funding, it sounds massive, but I think this is work in progress. You can never wait to have $1 billion in the bank before you start a project, but you keep accumulating, you demonstrate that progress can be made, and you generate additional resources. And you leverage other resources that exist to be sequencing things in other places. I really do welcome this kind of alignment with the EBP, with AfricaBP. I think in there we can leverage the resources to be a bit more efficient. And Eric, you mentioned the target of sequencing things only once. I would like to hear from you, what are the key parameters that you're building for us to say, okay, when we pick one species, we sequence it only once and we're sure that we have a good enough genome to move forward. If we can do that, that will be extremely efficient in using the resources that we hope to have access to. Thank you.
Okay, I can answer that and also give some suggestions or recommendations on some of those other questions. And so what are the parameters for generating, it's getting as close to a telomere assembly as of now, besides using those technologies that I mentioned? It's actually better to have a diploid individual. Better to have a more heterozygous individual than a individual that's highly inbred, because it allows you to separate the paternal chromosomes more easier that way. And This is part of my other answer there is going forward, getting to telomere. Someone asked about PacBio being not as easily available in Africa as nanopore is, and what's the future going to look like. I think to get to telomere, like with this telomere human genome, it's going to be a combination of PacBio and ultra long nanopore reads plus Hi-C, or maybe a few other things, to get it to sequencing once. So if you want to project a year from now in Africa, in certain sequencing hubs, what do you want? Or if you're collaborating with others, I will predict Hi-Fi, PacBio, nanopore, and Hi-C with the heterozygous individuals that you capture in your sample, not an inbred individual.
Thank you.
And in answer to some of the other questions about the database and the funding, what we did for the VGP is we had difficulty convincing governmental funding here in the US or other countries to give us money for the start of this project. What we were able to convince was our own institutions to give us the money to set up the sequencing hubs. And then what we did is individual scientists around the world contributed their own money to buy the reagents for sequencing of individual species. And that's how we funded the 260 species. That's how we got the money. So someone gives me the money for species in Africa and I contact, thank God, it says, okay, I have the money now, I need the species, or vice versa. So that's not the way I think it should be done in the long term, but it's been working that way so far for the VGP. And for the database, we've made an agreement with Amazon that this genome data, it's costly to store it. It's a lot of terabytes, right? And so, and if you're going to do many species, it's really costly. So we worked out an agreement with Amazon to store the data for free for the VGP project and anything that's associated with this GenomeArk website that we developed. They just want to be part of it. So they want to have the name associated with it. And so any vertebrate genomes, or even going beyond that, that you generate, if it's part of that, you can store it there for free. Now, Amazon, where they store it, it's distributed around the planet. It's not just here in the United States in terms of their servers. So what I would recommend is tapping into that kind of approach where you have, you can mirror all the data in Africa, but also have it back up somewhere else and collaborate with some of these companies that already have a lot of storage to begin with, like Amazon.
Yeah, thank you, Erich. I think Apollonia, this is something we made, Apollonia and Anne, kind of, I think we take on board, especially with explore other tech companies as well, Google and the rest. I think we had that conversation with the AS, basically. So there are lots of questions here. We have 10 minutes. So I'm just going to look at the other questions. Can Eric share his recommendation slide? So Eric, I'm wondering if you are happy to share, for us to share your slide. You send it to me and then I share it to the community.
I'll send you everything. I won't delete slides, and you can share it with the
community. All right, perfect. That works well. So apart from having databases, will you also conserve type species? Of course, the idea is to use the data for such efforts, basically. So someone asked if we are working with museums and taxonomies. So yes, the idea again is to identify all of these sample custodians, as Anne mentioned. So maybe Anne, or I'll give Anne or Roxana the opportunity. Roxana, do you want to comment on the taxonomy of this and the museum? So Roxana is the chair for the pilot committee, and she has recently put together the team yesterday. Just comment briefly on it, please.
So I see a lot of questions about the various details, and these are the same questions I've been asking when I first heard about this project. And the thing is that that's why we are developing this pilot project. This is to, just see what we can do and what kind of troubles we will get into. And then this is also to find the right people and allow them to work on this collectively. So we don't have these answers yet, but this is what we are working on right now. And you all can contribute this, of course. So I hope that we'll keep in touch, and then we'll inform you of any progress. And then you can also add your comments as well.
All right. Thank you, Roxana. I have Ahmed, could you please? Ahmed Hussein.
Yeah, thank you. Thank God my connection skipped a bit. I have a question for Eric. It was a very nice presentation. And as a student of bioinformatics, involved in a lot of genomic sequencing recently, I'm very curious about the comparison with the long reads and now traditional Illumina sequencing. And I have a question because from the presentation, it seems to be a very marginal differences that we see with these sequences. So I wonder, do you think it's a problem of the current bioinformatic tools we have to analyze the data? Maybe we don't have the high throughput that we need, or do you think it's a problem of the coverages that we from the sequencing data?
So what you said marginal differences between what?
Yeah, between the long reads, the high sec, and the ones that you talked about earlier, the differences between the sequencing with the compared to the traditional Illumina sequences.
Actually, to me, there are huge differences. Where if you're missing the promoter region for
in the
genes in the genome,
it's enormous. So it's enormous, right?
It's enormous, yes.
So that's why I'm really curious to know.
To me, it's immoral to do this is short read only assemblies now.
So I'm curious if you think it's a problem with the bioinformatic tools we have at the moment, or if it's a problem with the coverages.
The separation of haplotypes and false gene duplications is a problem of bioinformatic tool. The missing regions like GC-rich regions and getting through repeats is a problem of the sequencing technology. So the long reads, you get through repeats easier, so that answers that question. But the enzymes that they use to generate the long reads also read through GC-rich regions better than the Illumina-based enzymes or the Sanger sequencing-based enzymes. So those regions, the promoter regions, were never sequenced in the first place. So it's never sequenced, you can't even assemble it in the short.
All right, cool. Thank you, Ahmed. So I'm just going to treat one question on the chat box, and then we're going to go to the, I can see 3 hands up. Maybe let's just do this and then come back to it. So someone asked, what are the direct methods of collaboration to begin for involvement in laboratories? So as Anne mentioned, so maybe Anne, you might also want to comment briefly on this. The idea is for us to work with existing laboratories and infrastructure in Africa, basically, hopefully. And then for situations whereby we need PacBio Hi-Fi, which I think is very minimal at the moment, then we'll probably maybe want to connect with this. Do you want to add any more things to this, Anne? Or is that
no,
okay?
just that we're banking on having a regional coordinator for each region so that they are able to identify the specialists within the countries around that place. So we'll be using the experts that are already based in those countries. That's all I want to add.
Thank you, Anne. So I think I have like 5 hands up and we have about 6 minutes. I would just want us to go to the next agenda, which is looking at the monthly meeting, and then we can come back if we have a few minutes to spare. So on the next agenda, it's mainly the meeting frequency. So in order for this project to be successful, we need to meet. So the first thing and the key thing is meet, come together, talk, share ideas, and then we can go back to our various regional levels and implement this. So I'm basically thinking, as well as many members of the committee, that we should make this a frequent monthly meeting. So I just want to be sure that everyone is okay with this. And but the question is, when should it be? Should we leave it at 3 p.m. GMT +1? And this would be, I'm looking at the tip of Africa now, Nairobi. This would be 5 p.m. in Nairobi, Kenya. Is that okay for folks in the people in Nairobi? And should we move it to 2 p.m. GMT +1, which would be 4 p.m. Nairobi? But again, I'm looking at during this winter period when the people in Europe and the US kind of move backwards, then that would then make the people in Nairobi to stay in the meeting much later.
Thank God. You're talking about on Thursdays?
Yeah, on Thursday, there's this particular general meeting.
So keep in mind on Thursdays, once a month, or are you talking about meeting once a week or once a month?
So I'm talking about meeting once a month for this, for the AfricaBP.
Okay.
And yeah.
Okay, because there are 2 Thursdays in the month that the EBP meets and the VGP meets.
Okay.
So you're going to make sure you don't overlap those 2 Thursdays, because if you want some from the EBP and the VGP to participate and vice versa.
So I'm looking at us using the same, I think this should be, what Thursday? This should be let me see, what Thursday is this? Is this the second and third Thursday of the month? So this is the fourth Thursday of the month. So I'm looking at the last Thursday of each month, which is the Thursday before the EBP's meeting. So the EBP meets the first Thursday of the month, right? So I'm looking at the AfricaBP meeting on the last Thursday of each month, so a week before.
And the VGP meets on the third Thursday of the month.
There is no overlap there then. If we
No.
keep this as the last
Thursday.
Thursday. Yeah.
That's right.
Yeah, okay, should we keep it at the last Thursday of the month? The people agree with it. Then the question is time. Should we stick to 3 p.m. or 2 p.m. GMT +1?
Keep the same time until it doesn't work and we change.
All right,
It
perfect.
seems to have worked today, so
Okay,
don't
all right,
change
perfect.
it.
Okay,
Well,
perfect.
I don't know how many people it works for, but I don't think it worked for me. So I'm in, what do you call it? I'm in the western part of Canada, so this started as late, early as possible. So, but I don't know how many people that work for. But if we can go further, like go higher, maybe around 5 p.m.
So I think we'll need to take into account that we need to be able to have participation of African scientists, and the timing again in Africa. If we take it to 5 p.m., that would mean that someone in Nairobi, Kenya, will probably have to be on the call till 7 p.m. So okay. I think we need to take into account that what works for the African people in Africa. Then I think we'll probably see how we can work around that.
So we keep this time. We promise you some coffee when we meet.
Yeah. All right. Perfect. Thank you. So we'll stick to this time, and we'll make it a recurring meeting. Okay, Apollinia,
I think so.
You have a comment. So we're going to take just 2 more comments, and then we end the meeting.
Very simply, I just wanted people to use the chat box and send in as many questions as possible. It sounds to me that we can collect this and build a frequently asked questions sort of thing. How do you join? Do we have methodologies? I think if we do that, then we can post it somewhere for people to refer to. So I just encourage you to use it. Thank you.
Yeah, so let me just post a chat. I mean, you can also send us an email on AfricaBP @Google Groups, and I think that should be able to allow us to kind of respond to some of these questions. So Aneke and Sedik, I'm just going to give you maybe 30 seconds each, and then we close. Briefly, make a contribution.
Thank you. I just tried. This is a great chance for me. What I want to mention is I have not seen the wet lab aspects for. I mean, if we are planning to carry out genome sequencing, we need to consider how we can get high quality sample that's related to DNA extraction, those kind of stuff. So in the workflow of the organizational workflow, I have not seen those kind of how you guys handle the wet lab aspect to get high quality genome. Maybe it's not it's early part, but it's good to plan and kind of take into that consideration.
Yeah. So I don't know. Does any member of the chair wants to take this up, or the AfricaBP chair?
Yes, I had mentioned that we are looking to have hubs within the different parts of Africa. We are also very, very keen to have one technology, the highest long read technology for all the sequencing that we'll have on this. We are looking to develop the protocols for sample collection and genomic DNA isolation or RNA, in whatever the case might be. So these are still protocols that we're developing. We are going to not make new ones. We're going to use whatever is there already being used in the AfricaBP BioGenome Project. We're still in the beginning. We are happy to hear your thoughts on this, but definitely we'll have wet labs, we'll have sequencing labs, and we will try and do the best we can with the resources that we have. Thank you.
All right, thank you so much. I think we're on top of the time. Eric, you've had so many questions. If you please drop in your email address, then people could, maybe, I'm not sure how I want to do this, or I could send the questions to you, and then you can then respond to them. I would like to thank everyone for attending, and you will receive an email again from us for the monthly meeting. It's going to be a recurring meeting. We'll stick to this time. Thank you so much, and see you in July.
Thanks.
Bye-bye.
Bye. Thank you. Bye-bye.
Bye.
Thank you. Bye-bye.
Thanks.
Thank you. Bye.
Thank you. Bye-bye.
Thanks. Bye.
Thank you.
Thank you. Bye.
Thank you. Goodbye to everyone. Thanks. Bye, Prof.
Bye-bye.
Bye-bye.
Thank you. Bye.
Isidore, you are in the building.
Yes, Prof. I will pass by.
You're very resilient.
Yeah.
Dr. Ebenezer. Thank God. How you doing? This is Dr. Tillahan.
Okay, yeah, so I think Isidore, you maybe can stop the recording, and I'm not sure if people want to have a chat then.
It doesn't matter if it's recorded or not for me, but
yeah, I think because the meeting has ended, so
right.
It's just to save the storage space.
Yeah.